All right, let's take a journey together. We are about to dive into the most incredible, most complex instruction manual ever written. We're talking about the one that makes you, well, you. We're going to unpack your genetic code.
Have you ever just looked in the mirror and wondered, why do I have my dad's nose or my mom's eyes? You know, the answer goes way deeper than just saying genetics. It's a very specific set of instructions, a code really, written in a language that your entire body just understands.
And get this, the entire manual with every single one of those instructions is packed inside almost every microscopic cell in your body. I mean, how is that even possible? Okay, let's dive into this and find out.
So, let's start with the big picture. Imagine your body as this incredibly complex skyscraper. Now, before you can build it, you've got to have a blueprint, right? Well, that blueprint, life's instruction manual, is found in nearly every single one of your cells.
Yeah, this quote absolutely nails it. DNA contains all the genetic information needed to build and maintain an organism. It's not just the blueprint for how you're built, it's also kind of like the project manager, directing everything from your hair color to how your cells scramble to repair a simple cut on your finger. It's doing it all.
Okay, but wait a second. This brings up a huge question. If you stretched out the DNA from just one of your cells, it would be about 2 meters long. That's taller than most people. So how on earth does the cell solve this insane packing problem? The answer is just a marvel of biological engineering.
Well, the cell has this absolutely brilliant packing system. See, most of the time, your DNA is in a state called chromatin. And you can think of it like a messy, tangled charger cable just kind of thrown in a drawer. But, and this is key, when the cell needs to move or divide, it coils that DNA up super tightly into an organized structure we call a chromosome. It's the exact same cable, just neatly wrapped up and ready for travel.
So how does it coil so neatly? It wraps itself around these tiny little protein spools called histones. This basic unit, the DNA string wrapped around these histone beads, is called a nucleosome. It's the very first and most critical step in compacting that 2-meter-long manual into something 10,000 times smaller. It's incredible.
So if I were to ask you what a chromosome is made of, you'd probably say DNA, right? I mean, it makes sense. But here's the twist. A whopping 60% of a chromosome's mass is actually the protein packaging, those histones we just talked about, while only 40% is the DNA itself. That means the packaging is literally heavier than the instructions it's carrying.
So, we've established the packaging is super important. But now, let's zoom in on that precious 40%, the instructions themselves. To do that, we have to go all the way down to the molecular level, to the language of life itself, the famous DNA double helix.
Now, this iconic structure wasn't just discovered overnight. The story really started way back in 1869, but it was the absolutely essential work of Rosalind Franklin in the early 1950s that gave Watson and Crick the final pieces of the puzzle they needed to publish their double helix model in 1953, and biology was never the same again.
So what are the actual building blocks of this amazing helix? Well, each link in the chain is a nucleotide, and every single one has 3 parts. There's a sugar and a phosphate. They link up to form the long, twisting backbones of the ladder. But the most important part, the real star of the show, is the base, one of 4 chemical letters that actually write the genetic code. And this is maybe the most important rule in the entire genetic alphabet. The letters don't just pair up randomly. Nope. A always pairs with T, and C always partners up with G. This one simple, strict rule is the secret to everything. It keeps the DNA ladder stable, and even more importantly, it's what allows the code to be copied with incredible accuracy time after time.
Speaking of copying, let's talk about why this all matters so much. You see, the whole point of having a blueprint is to be able to make more of what it describes. So it's time to see how our cells use this code to copy, paste, and divide.
You know, we often picture cells just constantly dividing, but that's only a tiny, tiny fraction of the story. A cell actually spends about 95% of its life in a phase called interphase. And this is no chill-out period. It's a complete flurry of activity, where the most important job of all gets done, duplicating every single letter of its DNA instruction manual.
Okay, once all that DNA is copied, the main event, mitosis, can finally begin. Think of it like a perfectly choreographed 4-act play. In Act 1, prepare. The DNA condenses into those visible chromosomes. Then, in a line, they all line up perfectly at the center of the stage. For separate, the copied halves are pulled apart to opposite sides of the cell. And in the grand finale, finish, Two brand new identical nuclei form, and the performance is complete. So how does the cell manage to grab onto and separate those chromosomes so perfectly? The secret is the centromere. This is the crucial connection point holding the two copied halves together. It's also the handle that the cell's machinery grabs onto to pull them apart. Seriously, without this central hub, a clean and accurate division would be impossible. Now, your body actually uses two different types of cell division. There's mitosis, which we've just been looking at. That's for everyday growth and repair, making identical copies to replace old cells. It makes two daughter cells that have a full set of chromosomes. But for reproduction, the body uses a special process called meiosis. This one creates 4 totally unique cells, and each one has only half the number of chromosomes. This is absolutely essential for creating sperm and egg cells. Okay, wow. We have journeyed deep into the microscopic world of the cell. But why? Why does any of this matter? Let's zoom back out and connect all this amazing science to your actual life. Understanding our genetic code has, and I mean this literally, revolutionized everything. In healthcare, it's opened the doors to gene therapy and life-saving tests for inherited diseases. In forensics, DNA fingerprinting is a cornerstone of solving crimes. It's even transformed agriculture and allows us to trace our own family trees back through all of human history. And that brings us to one of the most profound, most humbling facts in all of biology. When you compare your DNA to any other person on this planet, it is 99.9% identical. So if we are all 99.9% the same, just think about that for a second. All the incredible diversity, the complexity, the billions of unique human stories that our world holds, they're all written in that tiny final 0.1%. And over the next few minutes, we're going to break down these simple, yet super powerful rules that decide how life passes its secrets from one generation to the next. To really get a handle on this code book, we first got to learn the language it's written in. So let's start with a few key terms. These are the absolute building blocks for understanding heredity. First up, heredity. This is the big idea, the whole process we're talking about, that beautiful, complex way characteristics get handed down from parents to their kids generation after generation. So what's actually carrying all those instructions? Genes! Think of it this way. If your entire genetic code is a massive cookbook, then a single gene is like one recipe, a specific set of instructions for one particular trait, like your hair color.
But you know how recipes can have variations, right? The same is true for genes. These different versions are called alleles. So for that eye color gene, you might get a blue allele from one parent and a brown allele from the other.
Now, some alleles are just a little more assertive than others. A dominant allele, we usually show it with a capital letter, is the boss. It will always show its trait. A recessive allele, on the other hand, only gets to show itself if it's paired up with another recessive allele. No bossy dominant one in sight. And this brings us to a really important distinction, kind of like the blueprint versus the final product. Your genotype is your actual genetic code, the specific combo of alleles you have. Your phenotype, though, is the physical trait you can actually see, like having brown eyes or curly hair. So how do we talk about these allele pairs? Well, if you get 2 identical alleles for a trait, say 2 for tall, you're called homozygous for that trait. But if you get 2 different ones, like one tall and one dwarf, then you're heterozygous. Okay, so this all sounds great, but how in the world did we figure all this out? Well, the story, believe it or not, starts in a monastery garden with a monk named Gregor Mendel. You might know him as the father of genetics. For about 7 years, Mendel patiently grew and tested something like 28,000 pea plants. Yeah, 28,000. His amazing findings, which he published in his paper, literally laid the foundation for all of modern genetics. His method was just so elegant and simple. First, he'd start with purebred parent plants. Then, he'd cross them to create what he called the first hybrid generation. And finally, he would let that generation self-pollinate to create a second one, and he tracked everything every step of the way. And what's wild is that he could actually predict what would happen. He used this simple grid, which we now call a Punnett square, to map out all the probable combinations of alleles that could pop up in that second generation. It was absolutely revolutionary stuff. So what did he find? Time after time, this predictable pattern kept showing up. For every one dwarf plant, there were 3 tall ones, a perfect 3-to-1 ratio. This was a massive clue that something predictable and mathematical was going on beneath the surface.
This observation led him to his first huge insight, the law of segregation. It basically says that even though you have 2 alleles for every trait, you only pass one of them on to your kids. And which one gets passed down? It's a total 50-50 coin toss.
But Mendel wasn't done. He was a curious guy, right? He started to wonder, what happens if I track 2 different traits at the same time? Not just height, but maybe seed color too. So he did what's called a dihybrid cross. He bred a plant that had round, yellow seeds with one that had wrinkled, green seeds. The big question was, are these traits linked? Does being round mean you also have to be yellow? And the answer was a huge no. He found totally new combinations he'd never even seen in the parent plants, like round green seeds and wrinkled yellow ones. And once again, it wasn't random. It all appeared in this beautiful, predictable 9:3:3:1 ratio.
This led to his second major discovery, the law of independent assortment. It just means that inheriting one trait, like seed color, has absolutely no effect on inheriting another trait, like seed shape. They're all sorted independently of each other.
Okay, pea plants are cool. I get it. But what does this all have to do with us? Let's bring Mendel's principles into the human world and look at a perfect example. How our biological sex is determined.
It's actually really straightforward. Females have 2 X chromosomes, right? Males have 1 X and 1 Y. Since a mom can only pass on an X chromosome, it's the father's contribution, either an X or a Y, that decides the sex of the child. It's a 50-50 shot every single time.
But here's the thing. These chromosomes carry way more information than just sex determination. The X chromosome in particular is jam-packed with genes for tons of other traits, and this leads to a special inheritance pattern we call X-linkage.
And this is where it gets really fascinating. For recessive traits that are carried on the X chromosome, males are at a statistical disadvantage. See, since they only have that one X, a single recessive allele from their mother is all it takes for that trait to show up. And that is exactly why conditions like red-green color blindness and haemophilia are so much more common in men.
It's honestly incredible to think that this all started with a humble pea plant. Mendel's laws gave us the fundamental rules of life's instruction book, opening the door to understanding everything from evolution to modern medicine. The real question for us now is, as we learn not just to read this code, but maybe even to rewrite it, what story will we choose to tell next?